Leading a startup team, whether you're delivering a sugar rush, stocking coffee, or getting a regular delivery of snacks, Office Depot has solutions that fit every startup culture, from getting those first business cards and stationery to ordering fleece pullovers with your new logo. To learn how Office Depot and the California Technology Council have partnered to bring you savings on all of these startup essentials and more, go to californiatechnology.org/member benefits. I'm Daniel Levine, and this is the Bio Report. The lack of diversity in genomic data has been an issue of growing concern. It threatens to limit the benefits from the massive investment that's been made to date to transform biomedical research, drug development, and the clinical care of patients. We spoke to Jonas Korlach, Chief Scientific Officer of Pacific Biosciences, about the problem, how it's being addressed, and the role advancing technology can play in gleaning greater insights from the genomes that are analyzed. Jonas, thanks for joining us.
Yeah, thanks for having me on.
We're going to talk about genetic sequencing, the world of precision medicine, and the issue of genetic diversity. I think it might be useful to start with the concept of a reference genome. What's meant by that term, and how is a reference genome used?
Yeah, certainly. So the genome is the genetic material of an organism, which for humans is about 6 billion DNA letters, or they call base pairs, and they stretch across 46 chromosomes. Those are in 23 pairs, one each from the mother and father. And a reference genome is the sequence of one representative individual or a composite of individuals. And the reference term means that the scientific community was striving for the highest quality with the currently available technologies to decode the genome and have the best representation of the actual genetic blueprint. And so thus far, the scientific community has produced reference genomes for human, for mouse, yeast, and a few other species. And so the reference genome serves essentially as a common coordinate system for annotation of functional elements like genes, and allows you to compare different individuals and also species. So in a way, it is like latitude and longitude, which is the common system to indicate position on the planet. And it allows you to talk about specific genes, how changes in those genes may cause disease, and the like. So it's a really crucial framework that allows scientists, geneticists to talk about our genetic blueprint and how it relates to phenotypes and how it relates to disease. And so it really, the human reference genome really provided a way for people to talk about the genome across the globe, like a dictionary with a language understood by everyone. And as a side note, another very nice side aspect of this, the generation of the reference genome by the Human Genome Project, was that it was a truly international effort. People from many different countries, many different ethnic backgrounds and diversity working together on a common goal. So it's a little bit different in terms of some of the other monumental achievements, like the space race. Apart from the space station, it's still very much a national effort paradigm. The reference genome and human Genome Project was really truly a global effort, and I think that was important because it really brought us together as a species and also highlighted that we're not all that different from each other. But now, I guess what we'll talk about is that the differences do matter and need to be considered in terms of diversity as we go towards precision medicine.
Well, recently there's been a lot of discussion about the need for diversity in gathering genetic information. What does the term diversity mean in the context of genomes? Is it what we think of diversity as being? Does it imply nuances we might not normally think of when we speak of diversity?
Yeah, so there's a lot of different aspects to diversity. The human population is a wide range of different types of diversity: cultural, phenotypic, meaning the way we look, and also genotypic diversity, what kind of genes we have in our genomes. And so often, of course, the three are connected. Culture is passed on from parents to children, and also the genetics. Your genes are passed on from the parents to children. And so genetics explains many phenotypic traits, meaning what we look like, and that's part of the reason why different ethnic groups look a little different from each other. And so the most critical type for diversity in precision medicine is genetic diversity. And it's important to note that we spoke about the human reference genome, but it's not a complete encyclopedia that's all human. In fact, the reference genome is currently a composite from about 70 individuals, but for technical reasons, one individual contributed the majority, about 70%, to that sequence. Another side note is that the reference genome in most places is just a single sequence, but as I noted before, every human being has two copies, one from the father and one from the mother, and those copies are, of course, not identical, because otherwise your father and mother would look the same, and they don't. And so even from that individual, the reference only represents about half what is actually present in that individual. But coming back to diversity, each individual human has literally millions of base pairs in his or her genome that are not found in the reference. And that's what I meant by the reference is not a complete encyclopedia for all humans. And vice versa. There's millions of base pairs that are in the reference that may not be in my genome or in your genome. And so, for example, there was a recent study from Sweden where they used PacBio smart sequencing to assemble from scratch two Swedish individuals, and they identified about 10 million base pairs present in these individuals that are not present in the reference genome. And so that has pretty important implications because for having effective personalized medicine, it is important to have as much information as possible of the DNA sequence in the human population be represented in the reference genomes and these population genome databases to make informed analysis.
Well, I'm wondering if you can expand on that. As we move towards this era of precision medicine that many people have been anticipating, what's the consequence of not having adequate diversity in reference genomes?
Yeah, so, the medical interpretation of a patient's genome involves comparing that genome to the reference genomes so that you can identify regions in that patient that may be disrupted or broken relative to these reference genomes. However, typically any region of the patient genome that does not match well to a region of the reference genome is ignored, and so it does not contribute to the analysis. But often these unique regions of a patient's genomes are really interesting and maybe important. And so if the reference genome is incomplete and doesn't adequately represent the diversity or is a poor match for the genome of that particular patient with that particular ethnicity, then the power to interpret that patient's genome is reduced.
Are there any examples you can point to how this lack of diversity plays out in the quality of clinical care or the ability to benefit at a patient level from the knowledge we have?
Yeah, there are quite a few examples. And so maybe the first example I want to mention is the interpretation of variants changes in patients' genomes that cause rare and presumably genetic diseases. Genome sequencing has been very effective at improving the diagnosis for these types of patients, but currently it is most effective for Caucasians and for Europeans who are best represented by the existing reference genomes and also the population databases that have been assembled. And so that means that individuals from other ethnicities more often receive a non-definitive answer. As one specific example, there was a recent study in a cardiology journal where they found that genetic tests for certain heart disease, cardiomyopathy, they were better able to identify these pathogenic variants, meaning disease-causing changes in the DNA, in the genomes in white patients than patients from other ethnicities. And so, as I mentioned, it's not just the reference genome that have this bias and are an overrepresentation relative to Caucasians, but the other associated data sets and databases as well that really help make the clinical diagnosis. So there are large databases. One is called EXACT, where over 50% of the entries are comprising Europeans. Historically, there has been a bias in so-called GWAS, genome-wide association studies, that's looking to identify disease-causing genes, and in which non-European participants represented only 19% of the individuals in these types of studies, despite the fact that over 3/4 of the world's population live in Africa and Asia. Another example, Africans in general have a lot more variation, and so they are, as a group, are much less well represented by the human reference genome. A second category where this is important is in disease risk. As one example, there was a study looking at the disease rheumatoid arthritis, and this was carried out by a Korean group of scientists, and they asked a simple question, and they asked, for the Korean population, how many of the mutations that had been identified in Caucasians, in European, by a previous study are relevant and important for predicting rheumatoid arthritis in their Korean population? And the answer was 0. None of those mutations and none of those associations played any role in predicting the disease for Koreans. And another disease risk example is in cancer. It turns out that cancer markers, so those are genes and mutations in genes that indicate a certain cancer and cancer risk, they do not validate in an ethnic group other than the cohort and the group of people where it was used for discovering these. And those are due to differences in the local genomic structure between the ethnic groups that are associated with those cancer risk connections. Another example is it has ramifications of potential drug discoveries. So because most genomic variation is both rare and population specific, you can miss important opportunities if you're just studying a single ancestral group. So there's one example, there's a gene called PCSK9, and it turns out that if you have mutations in that gene that make it basically inactive, it is causing low cholesterol levels and a lower risk for coronary heart disease. And that would have been missed if the studies had been restricted to individuals of European ancestry. Luckily, this was carried out on an African American cohort, people who had very low cholesterol levels, and so the researchers found that those African Americans had a particular mutation in that gene that kept these levels low and resulted in fewer heart attacks. And so this discovery now led to the newest class of cholesterol medications known as PCSK9 inhibitors. So that's a nice example of highlighting how it really, everyone benefits by looking at a larger diversity with important ramifications for drug development. And maybe lastly, if you limit your studies to a single ancestry group, that can also have consequences in terms of misinterpretation for clinical diagnosis. For example, there was a study that identified certain genomic variants in African Americans, and these were initially classified as disease-causing for another heart disease because they had used largely European ancestry samples. But then it was found that those were quite common in the African population, and so they're much less likely to be disease-causing because those people didn't have the heart disease.
Why does this problem of diversity exist? Is it cultural barriers? Is it issues of access and outreach? Is it a geographical problem? What are the factors that are at play?
Yeah, that's a complex, or this question has a complex answer. There are many layers, I think, and so let me try to maybe touch on at least a few of them. So I think it was necessary to start somewhere, with a single reference genome, and by that very nature, it was inevitable that the first reference genome would suit some humans better than others. And there are a few other reasons that the scientific community has yet to fully complement that first reference genome with more diverse genomes. The first is a matter of scientific discovery. It actually took quite a bit of time to recognize just how much genetic diversity exists in the human population and between the different ethnicities. The initial research only focused on the small differences, the so-called SNPs, or single nucleotide polymorphisms. Those are single base changes, so just one letter in the DNA alphabet in the sequence is changed. And that initially suggested that any two humans were 99.9% identical at their DNA genome level. However, more recent work using technologies like Pacific Biosequencing has shown that large so-called structural variants, those are larger changes greater than 50 base pairs or more, those changes more than double the previously recognized differences. And so a single reference is quite suitable for cataloging small differences. It's not very good at managing those larger differences. The second aspect is a matter of budget. As I think many know, is the Human Genome Project was very expensive, about $3 billion, and very time-consuming over 10 years across multiple sequencing centers to assemble the first human genome. And, it was difficult to convince politicians and others to consider amassing those kinds of resources for subsequent references. However, this has changed quite dramatically, and today, with the progress in the sequencing technologies, it can be done much more quickly and cheaply. So, for example, you can now get a map of the structure variants I just mentioned for less than $3,000 with Pacific Biosequencing, and that's comparable to what it takes to get at the SNP table. So it's gotten much cheaper and much faster to do this kind of research, and that really enables now to look at reference genomes for other ethnicities. The third, I think, is just, historically, much of the genomics effort was funded by wealthy nations, and so I think maybe understandably they focused first on their majority populations. But of course, it's important that we continue from there to represent minorities in the wealthy nations as well as citizens of less wealthy nations. A fourth, I think, aspect of why this problem of diversity exists in genomics is a cultural barrier with regard to awareness and education, and also some aspect of trust, in that previously I think there have been experiences by minority groups that have not been favorable, and so there's some suspicion about the value that they would get. There's some fear, I think, about the risks of what happens with the information and how that's being utilized in the future and so forth. So there's a cultural aspect that I think is really important and that also needs to be considered as well.
Is there some critical mass we need to achieve to reach an adequate diversity in the genomic data we have? Is there any consensus on what that might be?
Yeah, I think the ultimate goal for personalized medicine is that you have personalized reference genomes, meaning that the limit is really one genome per human at that kind of quality that the reference genome now provides. We're probably still a little bit away from that because the sequencing and the entire workflow to get from a sample to a reference quality genome is still fairly involved. So in the meantime, I think it's generally acknowledged that a good step forward is this concept of having a reference genome for each major ethnic group. And we have seen that there are many national sequencing efforts now that produce, let's say, the Korean personal genome project, making the Korean reference genome. There's Japanese reference genomes, the Chinese reference genome. I talked about the one in Sweden, and they're all working towards this goal, effectively generating a reference genome that's more appropriate for their population and really cataloging for the first time these ethnic differences that then play a role in what I outlined with regard to diagnosis, disease risk, drug development, and so forth. And many of those, I should mention, or most, if not all of those, were enabled by PEG biosequencing. However, I think the community wants to go further than that, and this is the concept of a de novo genome, meaning you put together an individual's genome from scratch, not relying on the reference genome, because, as I mentioned, there's an inherent bias. You take data from an individual and compare them to the reference. As I said, if it's not in the reference, you can't analyze it. So leading scientists have noted that ultimately we want to go to these types of de novo genomes, meaning we put each individual's genomes together from scratch. Tim Lupsky, a very preeminent researcher at Baylor College, said that bringing these de novo genomes into the clinic, that's the goal that you would really want to strive for, and he said that 3 years ago. Evan Eichler at the University of Washington said that recently, that if we still use the reference genome 10 years from now, clinically, that we failed. We need to understand these genomes for each individual from scratch and from bow to stern. And so I think these ethnically diverse reference genomes that I talked about, the Korean reference genome, Japanese, and so forth, that's a transition to ultimately having de novo genomes and being able to analyze those. And so I think we're going to get there. And these movements happen at different speeds in the research arena versus in the clinic and so forth, because obviously it's a difference of trying to make a discovery of how the genome works versus being able to diagnose an individual and then have some sort of treatment. Another aspect that's important, I think, and that's really coming to the forefront now, is the importance of phasing the information. What that means is that, as I mentioned, each of our genomes is diploid. That means you have two copies, one from the father and one from the mother, and it matters whether those mutations are on the same copy or whether you have, let's say, one mutation each on the two copies. And so with the previous so-called short-read sequencing technologies, you cannot see that, but it is accessible with methods now such as long-read PEG biosequencing. And so most in the context of diversity that we talked about, you can immediately see this with mixed ethnicity individuals. And so they may have the copy from the father and the copy from the mother representing different ethnicities. And so you need to be able to parse out which of the mutations come from the father and which come from the mother to really get to the best possible precision medicine program.
The National Institutes of Health has kicked off its All of Us initiative, which is hoping to sequence at least 1 million people in an effort to extend precision medicine to all diseases. What's being done in this effort to ensure adequate genomic diversity?
Yeah, absolutely, and it's been really great to see that there's such a strong focus on genomic diversity. So I really want to commend the NIH for making a strong point of that. And it's clear from its name, the NIH effort aims to represent all of us across race, ethnicity, sex, gender, sexual orientation, and so forth. And so inclusion is a very explicit goal of that study, and it will be measurable through these participant surveys. I understand that to help recruit representatives from all of these groups, the All of Us initiative does not require health insurance. It's entirely free to participants. The program has an All of Us journey across America to visit all regions of the country, educate citizens on how and why to participate, and really raise awareness and educate so that we have truly a representative spectrum of the American population, which, of course, America is the tossed salad of. I don't like the term melting pot because it means that you would mesh it all together. I like the tossed salad analogy where we try to really all live together and really enrich our lives through experiencing the diversity from the different cultures that make up the United States of America.
What other efforts can be made to improve the diversity in the genomes available for study?
Yeah, I mean, in addition to the genetic diversity of those subjects, I think it's very critical that use an effective technology to study those selected patients and genomes. And so there are some popular tools like so-called arrays and short-read sequencing that are really unable to study large parts of the genome. And of those technologies available today, PacBio long-read sequencing really provides the most comprehensive view of genomes. I think it's important to not just look at the quantities and look at 1 million people, but also look at the quality of the data that's being applied, of the technology that's being applied and the quality of data that then comes out, because otherwise you're just going to have to do it all over again. And so applying the best quality tool, I think, will ensure that as much as possible of the information of an individual genome is captured, and then that will really help with the statistics and the associations to really understand what causes disease versus what are common variants that may not cause disease, et cetera.
Well, we think of the issue of diversity being a problem of participation, but are there scientific issues here? How much of the gaps in our understanding of the genome and variation in genomes have to do with technology? Are there missing pieces in our understanding of variation that we need to back and fill with the technology that we use to read and assemble genomes has gotten better?
Yeah, that's right. I mean, so this so-called structure variation is something that was known for over 10 years, but because of the prevailing technology that was used, the short-read sequencing, it really has not been that accessible. And so over the past maybe 2 or 3 years, we've really seen a renaissance of applying mainly long-read PacBio sequencing to fill those gaps, fill gaps in the reference genome, elucidate these types of structure variants. And it's quite remarkable, in those ethnically diverse reference genomes, tens of thousands of new variants have been identified that are new discoveries that had not been seen before, ever, despite of large so-called 1,000 genomes projects and so forth that applied the short-read technologies. And so this is now happening at a larger population scale, and so we are in the midst of really catching up on filling the true set of diversity, of genetic diversity within the global human population. And that will fill the databases, and I think we will hopefully within 3 to 5 years have a much more comprehensive view of all the differences that exist across the human population and the different ethnicities, and then in turn provide a better understanding through cohort studies with different ethnicities what causes disease and what doesn't.
What's it going to take to have precision medicine truly define the way we treat and prevent diseases?
I think there's a number of aspects that have to happen. From a technology point of view, I think we need to be able to sequence and represent patients' genomes in a manner that captures all their variation. We need to generate the databases that can then be used, and that's not going to be, I don't think that's going to be one reference genome, and this is already starting where the scientific community is working on so-called graph-based references, which capture a lot more variation. It's going to take a lot more data, initiatives like the Million of Us is going to be foundational for then giving an accurate diagnosis. It's going to take education of the doctors so that when the clinical report comes back from a truly whole genome analysis, they know how to interpret that with regard to the particular mutations, with regard to the particular patient's propensity to metabolize drugs. One example is, and that's called pharmacogenomics, the ability for individuals to metabolize drugs is very different. Some are slow metabolizers, some are rapid metabolizers, and so the doctor has to appropriately adjust the dose for the particular individual, and much of that is genetics. And it's another example where the short-read technologies have not been able to really characterize that fully, and so there are now assays in development where they use PacBio long-read sequencing to really look at those genes and have a much better prediction of how those patients will break down the medicines that are being prescribed. And then I think there's a cultural shift with with an awareness and an acceptance and the proper regulatory environment that protects people from misuse of that information by insurance companies, by employers, and so forth. And so the ethnic diversity part is, I think, a large portion because, we certainly don't want to be in a situation where certain ethnicities are better served in the clinic, and there's better precision medicine for some ethnicities compared to others. That would be something like analogous to if you break your arm and you go to the doctor and the doctor says, "Well, I can't fix that because you are like the quote unquote wrong ethnicity." That's absurd, obviously, and we want to do everything we can to stay as far away from that as possible. And the programs like All of Us and others around the world are really striving towards making that a reality.
Jonas Korlach, Chief Scientific Officer of Pacific Biosciences. Jonas, thanks so much for your time today.
Yeah, thank you.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.